Hello everyone, welcome back to my series of videos for Homer2. This one's going to cover basically the big boss function of Homer2. This will be typically what you're going to want to run at the very end of your processing stream. I'm veering slightly from my delivery of video for this one because there's a whole lot of input and output variables which I wanted to show examples of. So this gives me a very boring PowerPoint presentation, but it will get the point across. So you even have little images of the various formats of each of the variables. So as we go through, it's just going to be very quick and dirty. We'll cover all the variables, both put in and out. Keep in mind some of the names from the comments in the function have actually been changed, and most of the input you really won't have to worry about because that'll be established during the processing stream. So you're not going to really have to individually put these in unless you're running the script through MATLAB. So the first matrix going in is going to be your Y matrix. This is just your data. It's three-dimensional. The rows are going to be each of your time points. Keep in mind those are frames. Excuse me, so it's number of seconds multiplied by sampling frequency, so it will be your frames, not in seconds. The columns will be your hemoglobin species, and then the third dimension is going to be the number of channels you have. Keep in mind, whenever I say hemoglobin species, that's going to be HBO, HBr, and HBT, which is oxygenated hemoglobin, deoxygenated hemoglobin, and total hemoglobin. The channels are going to be the combination of all the sources and detectors. Typically, it's every single possible channel, but you can also pre-prune this out to remove any unwanted channels. So either you remove channels because they're too noisy, or you remove channels simply because they weren't of interest. So they were, say, on opposite hemispheres of the head. This is what it looks like. If you pull it up in MATLAB, this is actually a picture pulled up of that. So if you notice the value here, you're looking at all rows, all columns on the first page, or the first dimension. So basically this is going to be all time points, all hemoglobin species, which is why you have 3 columns, because it's oxygenated, deoxygenated, and total hemoglobin. And then we're looking at the first channel. Keep in mind there's also a coefficient applied. So all of these values are going to be multiplied by this value here. And keep in mind also that these are going to be in millimoles. So the scale factor, or the coefficient here, you're looking at millimoles times that, which turns it into a slightly different unit, but listed as millimoles. The S vector, I know there's been a few questions as to how to make an S vector or exactly what the S vector is on the forum. It's basically your stimulus vector. So it's two-dimensional. It's just the number of time points and by the number of conditions. Again, the time points is going to be in frames, so it's number of seconds multiplied by sampling frequency. And every single, and the conditions will be, you know, like I have an example here. If you're doing finger tapping, you'll do left hand will be condition 1, right hand will be condition 2. So left hand would take up column 1, right hand would take up column 2. The vector is all zeros, or we can call it the matrix, is all zeros. The only place there will be a 1 is at the onset of a stimulus. Other than that, there's no offset notation, there's nothing like that, and that 1 will only be in the column of the event. So if the event is left hand, you'll see a 1 in column 1 at that time point. If it's right hand, you'll see a 1 at column 2 at that time point. This is what it would look like. So you can simply, I'll do a quick little video on it later, but you can easily make this in MATLAB. You can just create a vector of the correct number of time points, figure out what time point your onset was, and convert it from seconds to frames, and place a 1 in that column. But this is what it would look like if you notice here in column 2, at time point 1, which is 0, time point 0, you have onset of condition 2. At time point 8, I have an onset of condition 8. The T vector is going to be, it's just your time. It's literally just what time points were included. So when I say back on the S vector, when I say, you know, frame 3, I can actually coordinate that in a T vector as to what time point that is. and I'll show you that in a second. But the format of this is just the time points by the condition, so it's very similar. Or excuse me, I actually have this incorrect. It's actually just the time point. So it's actually one-dimensional. It just lists it down as it goes through. And here's an image of it. So I'll correct, in the PowerPoint that I present, I'll correct this to where it's only one-dimensional and just has the number of time points as rows. So here you'll see time point 1 is actual seconds 0. Time point 2 is 4/100ths of a second. So this would be, to convert to sampling rate, you would just, what is that? Multiply that by the sampling frequency, or divide the seconds by the sampling frequency. Math has not been my strong suit as of late. But you basically have the frames here simply converted into seconds. With the SD, this is going to be kind of a major struct in MATLAB. So it's basically a kind of a big box housing a whole lot of things. So the MATLAB will house your source positions, your detector positions. These are going to be in XYZ format. It's going to have the number of sources, the number of detectors. Lambda will be your wavelengths. Mesh list, which will actually go into a little bit more detail later. You have the source map, the xmin, xmax. You have a whole series of various variables within this struct that need to be put into play. I will actually not go too much into detail here, but I will do a separate video strictly on the SD struct because it is important, such as you have the spatial unit, which if you're ever using Atlas Viewer, it is important to note that you need the spatial units in millimeters, not centimeters. So all these I'll actually go into more detail in a separate video, but just know that this is a very large variable housing several other variables, which in themselves are multidimensional or cells or other variable types. The A auxiliary, or AAUX, is going to be all your regressors, all your auxiliary measurements essentially. So if you have a heart rate sensor or skin conductance sensor on the subject, or respiration belt or whatever they're called around the lungs, this will be housing all of those various data input. So the format of this is similar to the format of just about everything else here. The rows are going to be your time points, and the columns will be the number of channels, but these are specific to your auxiliary measurements. And keep in mind the time points will again be frames, not seconds. So this, you may have to figure out a way to downsample one or the other to kind of line them up a little bit more appropriately. May or may not need to do that, but that's something to consider. So tincAuto, this is going to be any motion artifacts that were denoted in Homer. So it's going to be just literally time points, and it's going to be zeros or ones. Anywhere where there's a 1 in this, so it'll look like your stimulus vector, but it's going to be one-dimensional. Anytime you have a motion artifact, it's going to be flagged with the number 1. Everything else will be 0. That's going to be used as whether or not to include it, or if we're later down the way deciding what we want to do with motion artifacts, we may actually remove that time point and some time points around it either before or after, just because it's unreliable. There's too much noise in it. So this is where we actually get to the beef of the function. It's the glmSolveMethod. So you have two options. One's going to be your ordinary least squares. If you choose number 1, you're going to use ordinary least squares. Number 2 will be the iterative least squares. There's a little bit more information on this in the comments on this, but basically for ordinary least squares, you're going to compare each and every data point to the model, and you're kind of doing the you're estimating the parameters of the generator model by best appropriating your model to the data. The iterative least squares, you're going to redo this over and over and over and over, and you're going to weight this, and you're going to actually find the absolute difference between the data and the model. Because you're having to iteratively do this, it takes more computational time than the ordinary least squares. It's actually suggested that if you're kind of just visualizing the data, or you're trying to get an idea of the data, you're going to use the ordinary least squares to save on time. And if you're looking for a little bit more beefy, or I think the most favorite word of everyone in this field, the most robust option, you'd use the iterative least squares, or number 2. The idxBasis, this is going to be which basis function you're going to use for the GLM. You have 4 options. 1 is consecutive Gaussians. 2 is a modified gamma function using a square wave. 3 is a modified gamma function and its derivative using a square wave. And 4 is a generalized additive model. That one I won't cover in this video. To be honest, it escaped me as to how to describe it. So I'm going to try to do a separate video on that to describe it a little bit more in depth and allow researchers a little bit better idea, who don't know much about it, a little bit better idea about it. But I will get to that on a later date. So the first option is going to be consecutive Gaussians. Best way to think of this is literally multiple Gaussian curves or bell curves just squished up against each other. You vary the width of the Gaussians, and you vary the spacing between Gaussians to create a shape. And then these shapes are, I guess you can think of it more or less average. So if you notice on our view here, we essentially have a bump here, a bump here. These would be essentially 2 Gaussians next to each other that are then merged into one. And whether or not that's completely accurate, it at least allows you a visualization of what's happening here. The input for this is going to be your standard deviation of the Gaussians as well as the step. The step is just, so the standard deviation is basically saying how wide the curves are. So just like in just general statistics, a larger standard deviation means a wider curve. Narrower standard deviation means a narrower curve. So it'll be taller and skinnier versus shorter and fatter. The step says between the medians of these curves, how much distance and time am I going to set between these? And your output ends up, of course, it's going to depend on the data, but it ends up looking something about like this. A modified gamma function is going to be it uses essentially knowledge of what the curve should be. So if you're doing a very well-versed experiment, that's a blocked experiment where you have certain amount of rest followed by certain stimulus, and you know the expected. So I'll show you the variables here: tau, sigma, and t. Tau is going to be the onset delay, or how long after the stimulus the curve is expected to onset. So typically that's a few seconds, up to upwards of 5 or so. And then sigma is going to be how wide is the peak up here going to actually be. So if I increase sigma, this width here will increase. And then t is going to be what is the duration of the square wave that I used to build this shape. So you use these basically parameters to figure out what the shape should look like, and then that's applied to the data. Next is going to be a gamma function with the derivative. So you use essentially the exact same variables put in. It's going to be tau, sigma, and t. Again, tau is onset delay, sigma is going to be the duration of the peak, and t is the duration of the square wave. But this will also include the actual derivative, or the rapid change of the function as it goes through. As you can see, you get a little bit more variety in the shape. Again, these are just generalized shapes. It may appear different. The data is going to deter it. I just wanted to include this to give you an idea of what the output would be. And if you change these values as well, the shape itself would change. You do notice that there's the initial downshoot, which would be typically seen. It may be slightly severe in this case, but you can figure out based on whatever data you have, whatever your expected values for that to be, you can adjust and best fit your group. The generalized additive model, I won't go into too much detail on this because I actually don't know too much about it myself. But you'll see the formula here that's used to calculate, and your input are going to be the p, q's, and t's, are basically all the variables used in the formula. The peak value, I believe, was going to be p times q, and t, I believe, was your was another square wave, but I'd have to actually adjust that. It could be the time. So don't think too much into this on this video. I'll get into more detail in a separate video and hopefully have a little bit more accurate information for you on this. The SD short separation threshold. This is basically stating what is my threshold if I'm using short channels, which I think most everyone here may have at least heard of them. It's basically a modality used to limit the distance between a source and detector so that you can measure very superficial measurements such as superficial blood flow or things like that. So you can eliminate it from your data, or you could use it for a number of other techniques that it's still kind of taking shape, but it's highly popular right now. But the basic idea is that if I put these sources and detectors close enough, I should not be measuring any brain. So most literature labels short channels below 15 millimeters. So that means the distance between source and detector is less than 15 millimeters. That means where the light and where the detector literally are touching the head, that distance is under 15 mil. The shorter you can get, the shorter measurement you'll get, and the better signal to noise ratio technically you're getting. You're getting much better signal because you're getting a lot more photons, but whether or not that is your signal of interest or not, or noise itself is another case. Oftentimes these are just used to regress out any superficial activity or what would be considered to be noise. What this function does is you just state in what am I considering to be a short channel. If I put 15 millimeters in, it's stating anything under 15 millimeters I'm going to include as a short separation channel, and from that I will treat that separately and differently in my analysis. This is going to be how you treat it basically. So flag SS is again short separation. So it's how to treat any channels labeled as short channels. So if I click 0, it's going to use the nearest neighbor regression. If I click 1, it will use the channel that has the greatest correlation to the signal. And if I click 2, it will average out all of the channels, the short channels that I do have. Typically, most individuals will not have many. Oftentimes, it's just one short channel. I personally have seen as many as 4, but I'm sure there's more out there. So using the average may or may not matter. Using the greatest correlation may or may not matter if you only have 1. If you have more than that, that's when you would want to actually take that into consideration. And if you don't have any, you really don't have to worry about this. The motion correct. So this is basically going to be anything that is noted as a motion artifact. It's asking what do you want to do with it. So if I click 0, I'm not going to do anything with it. It's a motion artifact, but we'll disregard it. This may have been taken care of actually separately in a previous function. So it may not be something you're too concerned with. Aside from that, though, I could also baseline correct it with epochs or between epochs. So if I mark it as a motion artifact, I can then basically you're essentially like kind of averaging out those around it as a baseline. So you're removing, say, 2 or 3 seconds of data, and you just put a flat line between it. So it acts as a corrective function for any motion that may have interfered with your data, and essentially the goal is to make it neutral. So it's not going to just create or destroy any unnecessary data. So now we're into the output. So that was all the input. Most of the input you're really not going to worry about, aside from the variables you choose within the GUI. Like the Y matrix itself is going to be moved into this just from your nearest file. But the output is actually quite interesting, because this is actually what you're not only going to visualize, but also going to utilize for any references. So first and foremost, it's going to be found in MATLAB. So once you get into MATLAB, you can load up your post-processed script, which is probably going to be called, whatever experiment.nirs, or I always have it as digpts.nirs, because I think that's for Atlas viewer purposes, but I won't worry about that now. But you can load it up. You'll find your variables pop up on the workspace, and you have a struct called prosResult, and this is where all of your output will be found. So the first output is in the comment section is called yavg, but it's actually been renamed to dcAvg. DC would stand for delta concentration, or the change in concentration, which is then averaged. This is the original data set, and it's now averaged by column. The overall average per column, and the format is the number of time points as your rows. And it's four-dimensional, so you have time points as rows. Again, you have 3 columns, which is going to be your HBO, HBR, and HBT. Then you have the number of channels as your third dimension, and then finally you have your number of conditions. So the actual appearance of that is going to be here, and you'll see we're looking at all rows, all columns of page 1, section 1, and whether or not that vocabulary is correct, we'll worry about that. But you have your scaling factor here, or your coefficient here, and then you have your values here. The next down is going to be your standard deviation of your matrix. It was renamed actually in the code as y average standard deviation, and then it was renamed in your output as delta concentration average, or dcAvg standard deviation. And this is the standard deviation across all trials. So again, the format is going to be four-dimensional, and it's about the same. You have time points by hemoglobin, and again, that's why you have the 3 columns. And then you have by the number of channels, and then by the number of conditions. So what you'd see here is these the conditions would alter as I scroll down through this, and obviously I don't have all of my data here, it's just a screenshot. So the conditions would change 1 through however many conditions I have. I think I had 3 or 4 here. Once I reach 4, then you'd look at channel number 2, and 1, 2, 3, 4, and then channel number 3, 1, 2, 3, 4, etc., and it goes down like that. The next is going to be the THRF, which is time hemodynamic response function. This is just another time vector. So again, my timing series was -5 to 35 for the block average window, or sorry, stimulus window, or whatever you want to call it. But what you'll see here is it's not exactly -5; it's -5.12, and then you have your 0 point here. So this is all the baseline data here, all the baseline time here, and it just depends on your sampling frequency what your -5 will actually be. These values are denoted in the input variable of t range in the processing script. So then you have your number of trials. This is just going to be, it's a column vector listing the event markers in chronological order. In my case, I had event marker 4 happen 3 times in a row, 1, 2, 3. It doesn't tell me when it happened, just in chronological order, not necessarily what time stamp. And then my 4th was going to be event number 6, then event number 5, and finally event number 6 again. So I had a total of 6 events occur during my experiment. Don't recall what my experiment was in this case. More than likely something ridiculous I was wasting my time on. But I have 6 total events, and there's only 3 differing events. The y new is going to be the same as y sum 2, and also the same as y residual, which was renamed delta concentration residual, a DC residual. So here it is. So again, there's been a few name changes throughout this, but we'll see if we can update that soon. This is going to be the residual model of the HRF. It's 4-dimensional. As you can see here, again, you have your scaling coefficient, whatever you want to call it. It's going to be number of time points as your rows, again, number of hemoglobin species, channels, and conditions. So again, we're looking at all the time points, all this hemoglobin, which again, we have 1 column, 2 column, 3 columns. So it'll be HBO, HBR, HBT. We're looking at channel 1, condition 1. Again, you'll run through all the conditions, and then move up, click up 1 on channels, run through all the conditions, click up 1 on channels, and you'll go through as your data set goes through. And also keep in mind you do have your real and imaginary aspects of each of these. Then you get to your betas. This is going to be, as I put, the meat of your output. I think I've said that twice now. These are just your beta coefficients. They're going to be per channel, per condition. They're 4-dimensional. The actual variable itself is 4-dimensional, which is 1 times the number of hemoglobin species times the number of channels and the number of conditions. So here you have essentially, we're looking at all rows, all columns, we're looking at channel 1 condition 1. Here's channel 2 condition 2, channel 3 condition 1 or 1. And then also keep in mind that these are, if you see, there's actually only 2 columns here. HBT, or the total hemoglobin, is not included on this. So that's a special note to include on this. This is only the the second dimension here is actually only going to be 2, not 3. And then finally we have the R, which is just going to be, I think most everyone's familiar with it. It's the Pearson correlation coefficient. Hopefully I got that right. And it's per channel, per hemoglobin species. It's 2-dimensional. So we have channels, which you see 1 through 18. This is the bottom of my data here. And I have 3 columns. So I have HBO, HBr, HBT, channel 1, channel 2, channel 3, and it goes down like that. So there we are. So this is going to be just a quick and dirty run through of the format, really more than anything, of the Homer deconvolve href drift ss function. It doesn't really go too much into depth as to how everything's done, which I do have notations on and I can do another video on. But I found that most of the questions really were more along format and how to either create certain variables or how to extract certain variables or where to find certain variables. So I hope this does help. The most notable point I do want to drill across is going to be that all of your output will be in your .NIRS file, so you will have to reload it into MATLAB, as I think the function is load. Then you put the name of the file into parentheses, including the .NIRS, and you load it as a mat file, which is an in parentheses -mat. Once loaded, you'll see all of the output in this pros result file. If you don't have some of the output, there's a chance that some of your input was missing. If that's the case, I would suggest putting a MATLAB stop sign on one of the lines of code, or the last line of code even, in this function, and you can actually scroll through the variables to see that everything is there or not there. Other than that, I hope this was helpful. I'll do a quick video next about just building a stimulus vector. And if you do have any requests, please let me know and I'll try to get to it as soon as possible. Thank you.